Okay, this is going to be a long video. Some of you have already been asking for a Unit 6 review, and since we've had virtual days leading up to the Unit 4 and 5 exams, I made reviews for those ones. And so for this exam, I thought I might as well do the same, especially because some of you guys are asking for it. And so since you're asking for it, I'll be polite enough to try to oblige. As you can see, a lot of notes up here. And guess what? There's like 2 of these slides, and so good amount of content. I'm going to try to start from the very beginning, make my way through. This should be a good companion video to help you get through the review homework assignment for the test. Okay, so I'm going to start with the very front up here, top. I'm wondering if this will be mirrored, top left or top right. I don't know if this is mirrored for you guys. So I'm going to zoom in up here. Here are some of the main differences I want to go over real quick before we hop into anything else. Genotype versus phenotype. Those are 2 vocabulary words that we have in our notebooks that are very familiar, and so sometimes it's easy to get them confused, okay? I try to think of really one vocab word, and that helps me remember which one's which. So we have genotype. I think genotype just means the type of gene you have, okay? And we talked about this word alleles, which is the variation of a specific trait you have due to a variation in a gene. So different type of gene. Alleles that we've been talking about are different letters. So we have big B, little b. Those are alleles. Alleles equate to letters. So if you see a letter, that's an allele, at least if it's just by itself in a genetics term. So when I see a big B, little b, I know that's talking about a specific gene. That's genotype. Okay. Phenotype is based off genotype, so they're kind of companion vocab words. Phenotype is how you look on the outside. Okay. So for example, big B, little b. I said someone who has a big B, little b for a specific gene, like hair color. If you have a big B, you're going to be having brown hair, okay? So phenotype is how you look. Genotype is your specific gene combo, your allele combo. Next up, we got homozygous versus heterozygous. So I'm going to... my head's just in the way. Okay, homozygous. The root word within that vocab word right there, homozygous, is homo. And I always ask you guys in class, what does homo mean? And there's other words that you've heard with that root word in there. And homo means the same. So that means you need the same exact allele, same exact letters. So homozygous, you can either be homozygous dominant, which means you have 2 dominant alleles, or you could be homozygous recessive, which means you have 2 lowercase alleles, okay? But homozygous means you need the same letter twice, exactly. Capital, capital, lowercase, lowercase. I know the letter B is the same, whether it's uppercase or lowercase, but when we're talking about alleles, it's not, okay? A big B is very different from a little b, dominant versus recessive. Heterozygous, the root word hetero there, means opposite or different. And so that means we're going to have 2 different alleles. And if we have same as big B, or little b, well, to be different is to have one of each. So that means we're going to have a big B, a dominant allele, little b, recessive allele. So what's the difference between a dominant and recessive allele? Because I've been bringing up those terms, and you guys, really based on this video alone, would think that dominant means capital, lowercase is recessive, okay? That's kind of the right track, but we need a little bit more than that. So dominant versus recessive. If you have a dominant allele, okay, a dominant gene, like big B, that means you're going to have the dominant trait. So if you get one big B or one dominant allele, you're going to have the dominant trait as your phenotype. If you have 2 copies of the recessive allele, so for example, little b, so if you have a little b, you're going to have the recessive trait, okay? But you need 2 recessive alleles, 2 little b's to show the recessive trait. Because that big B, the reason it's called dominant is because it silences recessive alleles, recessive genes. So anytime you get a capital dominant B, that means it's going to take over. That's the trait you're going to have. I said in this circumstance that brown hair is dominant, and I made blonde hair recessive. So if you want blonde hair and it's recessive, you need 2 little b's, just like this. You need to be homozygous recessive. If you get 2 big B's or 1 big B, you're going to have the dominant, okay, phenotype, which is brown hair. Last but not least, we'll be tackling this a little bit more on the next slide, protein versus enzymes, okay? And proteins are these things we use in our body that we build ourselves, okay, using a structure called ribosomes. Again, talking about that in the second slide. But proteins have many functions, okay? One example is signaling, okay? Your proteins can signal to the cell, we should start building this up, or we should start breaking this down, or we should start changing the cell in some way. We should start splitting the cell. The proteins have many jobs that relate to structure, okay, holding the cell together. For example, when we are doing cytokinesis and the cell is actually starting to split, what happens is down the middle of the cell, around the perimeter of the cell, there will be a protein ring, and that protein ring will start squeezing until it kind of splits the cell down the middle, and now we have two cells, okay, like pinches the middle. So we do have proteins that help with structure, support the cell, or change the shape of it. We have proteins that act as transport. They essentially act as a big protein orb, and they'll shove things inside there and either transport those contents to other parts of the cell, or they may transport those contents within this protein ball to another cell. This protein transport ball, okay, may leave the cell and go somewhere else in the body. Okay, that's pretty normal. Number 4, proteins can break things down, which is an example of our enzymes. Okay, enzymes are proteins, but not all proteins are enzymes. It's the very same situation of all squares are rectangles, but not all rectangles are squares. I think I said that right. Same thing with proteins and enzymes. All enzymes are proteins, which enzymes are things that break down usually chemicals, and they speed up chemical reactions. But not all proteins are enzymes. Other things proteins can do, motor function, which just means movement. They can move cells or move things around the cell, kind of helping with that transport. Another thing is storage. We store things within these proteins just to store them for later, or sometimes we store them and then we inject in this protein like an acid, and then we break whatever's down. We break what is ever in that protein down, and then it's destroyed. Okay, that's called a lysosome. These are a lot of different examples of what proteins do, but proteins are really everything that has a job, movement, or often appearance. Okay, a lot of people don't think about the appearance part when it comes to proteins, but proteins are very instrumental when it comes to the human body or any living creature's body. Okay, and they're tied to enzymes because enzymes are proteins, but just because you're a protein doesn't make you an enzyme. If you're a protein, you can do all these other jobs, but you may end up being an enzyme, but not guaranteed. Okay, so let's go to some other material over here. Lovely. So we did some levels of Zoom. That was an assignment we did in class. Some of us did it together. Some of my classes did it separately in their individual teams. That's fine. But I want to make sure you guys get the right stuff. So up here, I have 2 chromosomes. Okay, and I want you guys to ask yourselves, how many chromosomes does an individual have? Does a human have? Do I have? Do you have? How many copies of each chromosome do you have? And when you guys think about that, there's a couple of wrong answers I don't want you to say. A lot of people say, oh, we got 46. No, that's wrong. You have 46 chromosomes in total before you replicate them for mitosis. Some of you may say 23, and that's getting closer, but that's because you get 23 from mom and 23 from dad. So that means you have 2 copies of 23, and so I said the answer right there. How many copies do you have of every single chromosome? You have 2. You get 1 from mom, 1 from dad. That's what I'm showing on this board right here. This, for example, is one of my parents. Okay, this is one of my parents I'm showing on the board. And so they have a specific chromosome pair. Okay, they have big B, little b. I said big B is brown. That's dominant for hair. Okay, and little b for blondes. They carry it. Okay, so since they have one big B, one little b, that means they have 2 different alleles. They are a heterozygous individual, at least when it comes to hair color. Okay, so on these 2 chromosomes, I pointed out the location of these 2 genes. Okay, these genes for hair color. The specific location of a gene on a chromosome is called a locus. Okay, so if I ever say locus on a test quiz or in class, I'm specifically referencing just that one location of that gene. Okay, and so I chose this location for the hair gene. Next, we're going to look at the gene level. So what I did is I zoomed in on this little stretch of bar here on each of these chromosomes. I zoomed in to see what the code was. And this was one of my parents' genome. ATG, ACA, TTT, ATC. Okay, so that's one of them. And then their other chromosome had ATG, ACA, TTA, ATC. I want to see if you guys can spot a difference between those two strands right there. I think the difference is pretty obvious. Well, if you look, this first box here, this first triple code set, this triplet, are the exact same ATG on both these strands, okay? So, nothing different, they are the same. Next I have ACA, okay, ACA, the same. Then I have TTT, TTA. Now that is not the same, okay? And then if I look right here, ATC, ATC, the same. So it looks like my one difference is between this box here, okay? And so that means that's probably resulting in the different variations in that gene that's causing the two different variations of the phenotype, blonde versus brown hair. But my question is, which one is big B, which one is little b? Because I didn't tell you guys that. And just looking at this, you shouldn't be able to figure that out, okay? ATG, ACA, which one is dominant, which one's recessive? Is it the one that looks more dominant? Because personally to me, I think TTT looks more dominant. So should I put a big B up there? No. Take away my degree. This man went to stupid college, okay? The real way to figure this out is, for example, looking at a child, okay? And if we look at this child, we can try to figure out, okay, based on this trait that they have, maybe we can figure out which alleles they got, which letters they got. So let's look at me. When I was a child, I had atomic blonde hair. And I'll show you it right here, because I think this is an adorable picture of me. Ignore her, that's not me. That's me, okay? Super blonde boy. My sister was also super blonde. Anyway, so blonde boy McGee, he had TTA right here and TTA, okay? So in this one box, he has TTA, TTA. We know blonde is recessive, so we should know that both of these strands are little b, little b. So now, based on this strand up here, what did my parents have? Did they have big B there, little b here? Let's see. So, whichever strand has TTA means that is the recessive strand. And so, right here, I see TTA, so that means this strand must be my parents' little b, and this must mean that this is the big B strand, the dominant strand, TTT. Based on my guess before of TTT looks strong, well, guess what? It's the more dominant trait, so that was a lucky guess. Very good for me, okay? So, recognizing DNA strands and looking at them and figuring out which strand may be dominant, which may be recessive, okay? But we're going to zoom in just a tad bit more to focus on this one last bit here. So, DNA level. We see that this entire box, TTA versus TTT, those are different, but it's not all the letters, it's not all the code, your nitrogen bases are your A, T, G, and C. It's not all the nitrogen bases in this box or this box that's causing the change. Realistically, it's only one letter that's changing. I have TTA versus TTT. This last letter is changing, and anytime you have a change in the DNA, so let's say, for example, all humans throughout time had TTT in that specific gene. So everyone had brown hair. And then someone got a mutation, and that led to TTA, blonde hair, okay? That is a change in the DNA. Sometimes that leads to a change in a phenotype, a specific trait that you show. Not always. Not all mutations lead to an external change. A lot of mutations don't do anything. But this one seemed to do one thing, which is change hair color. And there's one important thing I want to point out here. The last letter was changed, okay? Sometimes the first letter gets changed, sometimes the second letter gets changed. And you'll find this out when you look at this codon sheet. But you'll realize that if you change the first two letters of any codon, which a codon is just a set of 3 codes. So if I say codon, C-O-D-O-N, that just means I'm referencing 3 letters of code next to each other. So this codon, these 3 letters, if you change the first 2, you often don't change the resulting protein that's made. What you usually make is the same exact protein. But they found that if you change the last code in a codon, that actually will often change to a slightly different protein, okay? And when this makes a slightly different protein, we'll get a differently expressed trait, okay? So instead of having brown hair for everybody, this one letter change at the very end, switching to TTA, now we have some people with blonde hair being developed, okay? So last letter usually causes a change, okay? Mutation, a change in the DNA. Last but not least, I want to bring up this one point here. Proteins, okay? I brought them up earlier. Proteins do a lot of many jobs, and sometimes they make things called enzymes that break down chemicals and speed up reactions and sometimes build up chemicals. These proteins are made of these things called amino acids, okay? I'll go over a little bit more about amino acids on this codon sheet. This codon sheet will actually tell you, based on your 3 letters, which amino acid is going to be added to your protein. And some proteins are made of a couple amino acids, just maybe 2. Sometimes they're made of more, so maybe I have a 3rd or a 4th or a 5th or a 6th amino acid. And it's going to be a really long strand of amino acids, and then I make my protein. Okay, so amino acids made of protein, I'll talk more about that in a little bit. Last but not least, I want to do an example of pedigree. I find pedigrees really fun. They're just like little puzzles. I find them like the same type of enjoyment as Wordle. Okay, so here's a pedigree, and we're tracking blonde hair within a family tree. Okay, and this one is similar to mine, except I don't have these 2 siblings. These ones don't exist in real life. It's just my sister and I. But I wanted to add some hypothetical siblings in my life. Actually, I could argue those are my step-siblings. There we go. Awesome. Done. Even though that doesn't work at all. So, we assign big B is brown hair, little b is blonde hair. Well, it says that right here, my mom had blonde hair, so she must be little b, little b. My sister had blonde hair, so she's little b, and I had blonde hair, so that means I'm little b, okay? Now the question is, what was my dad? Well, my dad has brown hair, and so he must just definitively have a big B. But then the question is, does he have a second big B, or does he have a little b? Okay? And there's a really easy way to figure that out. I can almost instantaneously come up with an answer. But to show it, I'll use this Punnett square. So what I'm going to do is I'm going to put my mom's little b's on the side. So little b, little b. I'm going to put my dad's one big B here. So, based on the pedigree, I know I need to end up with offsprings with little b. So I'm just going to put a question mark here. I'm going to leave it blank. Okay? And so, big B, little b, that means in this box right here, I'll put one dominant and one recessive B. And that will be the same case right here. Now you need to ask yourself, big B, little b, what does that get you? Blonde, brown hair, what does it get you? And you should be telling yourself that it's going to get you brown hair because there's a dominant big B, okay? So this is a heterozygous individual. Now, what's going to be in these ones? How can I figure out what my dad's second allele is? Well, I can just look at what the kids are. Well, we have a little b, little b for sure down here in my sister and I. And so that means at least in one of these boxes, I need a little b. And the only way to get a little b down here is to have my mom pass on a little b, but I need my dad to also pass on a little b as well. If he passed on a little b there, that means he's also going to pass one here. So there's going to be 2 little b's there. Okay? So now I figured out what my dad is, but what are my hypothetical siblings having? What's my other older sister have, and what does my younger brother have? Well, the answer's already in front of me. My sister and I got little b's, okay? And so I know they don't have 2 little b's because they're not blonde. So they don't have this. And since my dad has a big B, little b, and my mom has a little b, the only other outcome is big B, little b, or big B, little b, okay? Which are the same thing. So that means both of my siblings must be big B, little b, heterozygous. Well, my mom, my sister, and I are all homozygous recessive. Okay? So that's some practice with Punnett squares. There's going to be good practice on the review. I'd recommend taking them seriously. Punnett squares, I think, are easy if you practice enough. But if you don't take them seriously and you don't practice them, you're going to have a hard time. Make sure you know those terms. Homozygous recessive, heterozygous, homozygous dominant. If I use that term on the test, I'm expecting you to know that homozygous recessive is 2 little letters, okay? Or heterozygous is 1 capital, 1 lowercase, okay? I'm expecting you to know that. So that is the first part here. Oh my goodness gracious, wasn't that a lot? That was like 21 minutes of content, but don't you worry, there is more content right now. Who cares about an episode 2 when you can just make a humongous first episode and make that the only episode? It's like a movie. So, let's go over here. Protein synthesis. Okay? We talked about this a little bit already, and so I want to spend some time talking about some more just so we get some good old-fashioned practice. I'm going to move this down here just to help me out just a smidgen. So protein synthesis. What does synthesis mean? Can you zoom, please, and thank you? There we go. So synthesis means to make. So the process of making proteins, okay? And to make anything for a cell, for the human body, whatever structure we want to make or whatever material we want to make, we need to reference our DNA because our DNA has the instruction to do everything. It's like Googling something. Okay, it's like the cell is like, shoot, guys, we need more transporters. We need more builders for the cell. We're about to grow a whole lot, and we need to move things around and build things. How do we figure out how to do? Can someone go up to the DNA and just like figure that out for us? Can someone go read that and come back with the information? Okay, so that's what we do. So inside the nucleus, we do a process called transcription. What it means to transcribe is to write something down, but often write it down in another language. Okay, for example, if you read the fantastic, critically acclaimed, best-selling book, probably the most famous book out there, World War Z by Max Brooks. If you were to read that in English, and someone's like, "Wow, I'm a huge fan of this. Oh, I have family members that only speak Italian. Well, I better write this in their language so they can read it." That's called transcribing, okay? So how do we transcribe? What we do is we take the DNA strand and we're going to break this thing open. So we have these 2 strands and they're bonded together, 1 strand, and then the second strand's down here. We're going to split those open using an enzyme called helicase, okay? Helicase is an enzyme, and helicase, what it does is it breaks open helixes. That's why we called it helicase. Often, we name enzymes after the things they break down. For example, lactase breaks down lactose. Helicase breaks down helixes, okay? So we use helicase and we break it open. And then we look at one of these DNA strands, for example, the top strand. And what we're going to do is make a new strand of genetic material, okay? And you may be asking yourself, oh, so we're going to make like more DNA? Is this just DNA replication? And no, it's not. But it's pretty similar, actually, okay? So during DNA replication, we use this enzyme called DNA polymerase, okay? So DNA polymerase is one of the few enzymes that we bring up ever, where it doesn't break things down, it actually builds things up. We make new material out of it. So it's one of the building enzymes. And so DNA polymerase makes more DNA during DNA replication. What we're going to build is not DNA, we're going to build RNA. Okay, so DNA is deoxyribonucleic acid, RNA is ribonucleic acid, very similar names, but just slightly different. Okay, and I'm going to answer the question, why are we going to build RNA? Why don't we just make a new DNA? So what RNA polymerase does is it comes and reads the DNA code, so it comes through here. Let me move this up. Bring to the front. So it's going to come through here and read the code, and as it reads the code, little by little, a little string of mRNA is going to be coming out its back end, and it's going to be binding each of these letters to the corresponding letter that matches. Okay, so RNA polymerase will make this RNA strand, mRNA strand, okay, and I colored it green so you can see the difference. So RNA is essentially a different language for DNA, okay? It's almost the same stuff, but not quite the same in a sense that we keep it in the nucleus, okay? I brought up that transcription happens in the nucleus, and the reason DNA is in the nucleus is to protect it, okay? We don't ever want to release DNA out of the nucleus because it's very dangerous with DNA. The DNA can be broken down. And so what we do is we make like a, lack of a better term, like a cheap copy of it. Okay. If this copy gets broken up, we're not upset about it because we expected it to break. Okay. So RNA, we use it usually, okay, as a messenger. Okay, so messenger RNA, that's what the M stands for. So mRNA is a messenger. We use this messenger and we take this strand and we're going to send it out of the cell. Okay, and then we're going to reform these two DNA molecules back together, seal them up, okay, and we're going to send the message on the way. This message is going to make the instructions to make whatever that the cell needs, whether it's like we need more builders. Well, now this little strand here is the strand that tells it how to make more builders, okay, or transporters. But one last thing I should point out is if you look at these strands, you should see right here, actually let me put these back together, you should see our normal matching scheme, okay? Thymine, which is T, binds to adenine, which is A. So you see your T's go with your A's. A's with T's, T's with A's, T's with A's, A's with T's, okay? Then we have our C, cytosine, and that bonds with our guanine G. So we have C's with G's, G's with C's, G's with C's, okay? That's pretty normal. You guys have seen that in Unit 5, okay? And you've seen it during this unit. When we make RNA, the one main difference of RNA that's easy to point out off the bat is we don't use one of the letters in DNA, okay? So here are the rules. DNA, we bind A with T, and we bind G with C. But the biggest difference is RNA doesn't use T's, okay? Doesn't use T's at all. Doesn't use thymines. It replaces thymines with U's. And so the U acts essentially like thymine. So uracil, U, is pretty much the same as thymine, T, okay? They act the same. So instead of just a T, we just replace all of our T's with U's. So A binds with U, G binds with C, okay? That's a simple rule. And if you actually look at the opposite strand of DNA, I keep doing that. There we go. If you look at these two strands, you should see that these strands are actually the exact same, okay? That's making sure that you do the right work. I have an A here, 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 a G, C, C, A, this is slightly off kilter, A, G. The only thing that's different is the T's. The T's just get swapped out with the U's, okay? So it's still essentially the same message, but RNA uses U's, okay? So all of this happens, all this transcription happens within the cell. Or not the cell, the nucleus, okay, to protect the DNA. And then what we're going to do is send this mRNA out of the cell, and we're going to send it to this guy right here named the ribosome, okay? What's the ribosome's job? The ribosome's job is to, send this to the back, the ribosome's job is to make proteins, okay? And when we make proteins, proteins do a lot of different jobs, so pretty much anything that does a job in a cell, more often than not, is going to be a protein that does said job, okay? And if you make a protein, that also usually makes a specific trait that you can see on your body. So how does a ribosome work? Well, a ribosome is actually going to grab onto this mRNA, and it's going to start reading it. And so what I'm going to do is shrink this down just a smidgen, so it can start reading parts of the code, okay? So it can start reading parts of the code. And so right here, I have beginning of the code. AUA is in this first open spot here, okay, in the ribosome. This is the part where your codon sheet is going to come in real handy, okay? So for every 3 letters of RNA you have, that will make a specific amino acid on the outside ring of this codon ring. I like to use the codon ring. I find it very easy. So the first letters here that are going to be used is AUA. That's going to be the one in the first, okay? So then I just look at here, A, U, A. And what does that make me? AUA makes me isoleucine. 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 Very good. So that makes isoleucine. So what are we going to do? Well, that means I'm going to grab a isoleucine amino acid. So isoleucine is an amino acid, abbreviated as iso. Okay? And that's going to come on right in here. And I want to see if you guys know anything. Is Mr. BB doing the right thing right now? And usually if he brings this up, that means he's not. Okay, this guy, again, went to stupid college. Okay, dumb man, dumb. Okay, no, Mr. BB's not doing the right thing. I don't want that right now. Okay, why don't I want that? Well, when we're doing this process outside the nucleus, taking a ribosome and having it read mRNA to make a protein, this is the process called translation. Okay, and translation just means we're translating. I know translating makes it sound like we're changing language, which was how I described transcription, so my fault. But translation is when we're taking DNA material and translating it into amino acid protein material. Okay, we're literally making a new material, not the same. Okay, it's no longer genetic material. It's not DNA or an RNA. It is now a protein. So, what did Mr. BB do wrong? Well, in every set of mRNA ever, you need to start with the start amino acid, okay? And if you look on here, the start amino acid for every single protein we're going to make, we need to start with the amino acid A, U, G, methionine. So we need to find AUG in our strand, and that's when we're going to start adding amino acids on there. So, let's see. Do we have an AUG? Well, we have AUA, so that means those three letters are useless. I'm not personally going to be adding any amino acids to my protein from that. If I look at the second bunch, I have AUG. Oh, ding, 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 very good. We found it, okay? AUG is the start signal for the ribosome. And so what AUG tells the ribosome is, hey, when you get AUG in here, AUG, we're going to start actually finally to build our protein out of amino acids. So, as I said earlier, AUG is the amino acid methionine, okay? And this methionine is going to come on in. It's going to ride on this structure right here. This structure right here is called a tRNA, okay? The T in this RNA, so it's a type of RNA, so we have messenger RNA, which gets sent out of the nucleus to go make proteins. It's a messenger. It's like, hey guys, let's make this, and they use the mRNA to make it. The tRNA stands for transfer RNA, and I will describe why it's called a transfer RNA in a second. So, we're going to bind this tRNA into our AUG, okay? AUG. And to bind it properly, we need the reciprocal letters it binds to. So AUG, we're starting with an A, so A binds with U. So we have a U here. The second letter of AUG is U, and so U binds with A. So we need an A. And then last but not least, AUG, the third letter is G, so we need a C to bind to it. So this tRNA that has UAC is going to bind on, and it has this amino acid called methionine on there. Okay? Then it's going to essentially scroll down, move this DNA, or this mRNA down, and it's going to move the tRNA to this new spot in the ribosome, okay? And this AUG is still bound on, but this section's about to move out of the ribosome, and we're going to scooch more of the mRNA in. So we want to keep this amino acid up here, methionine. So how are we going to keep it so we can start building our protein out of these amino acids? Well, we need to figure out what is the next thing we're going to bind to. We have a CCU. So if I use this handy dandy codon sheet, our CCU, I know it's hard to see, but this is a C, a U, CCU, that's going to give us proline, which I just abbreviate as pro. Proline is going to come right on in, and it's going to bind right there, okay? But now we got two of these tRNAs in here with these amino acids attached to them. So how am I going to start building my protein with all these amino acids? I got to combine them together. And so what happens is, the reason these little structures are called tRNAs is they're called transfer RNAs. They're going to transfer that little circle of amino acid up at the very top. They're going to transfer that amino acid onto the next tRNA. And so this methionine right here is going to move onto this proline, and it's going to connect off of it like so. I'm going to have another orb here, and the methionine is going to go, let me draw it in black so it's easier for you guys to see. This methionine is going to go right on to the end of that, and this transfer RNA right here is going to lose its methionine, and this tRNA is now going to leave, and now these two are going to bond together. So let me make sure I bond these together. Group. And now they're going to move over here as the DNA molecule scrolls on down. Sorry, RNA molecule. Keep accidentally saying that. It's that tricky. The teacher makes mistakes all the time. So now our CCU is bound, and the proline and methionine are on there. And we're going to keep on going. So next we have we have AUA. So we have AUA. And so let's see here. AUA, that gets us isoleucine. So that isoleucine is actually going to be handy at the end of the day. So it's going to bind right here. And now my proline and methionine are going to hop on and join the isoleucine over here. And they're going to let go off of this tRNA. And they're just going to keep binding on to each tRNA as they move on through. Now this tRNA is going to move out. And now we have our proline here and methionine here. And now they're going to be all sorts of bound together like so. And now they're going to move over here as we scroll this down. And last but not least, we have UAG. If I look, UAG, that's the stop amino acid. But there's no actual real amino acid that codes for stop. What that actually tells the ribosome is, hey, that is the stop codon. There's no amino acid for that. It's just telling us to stop. So that means we're done making our protein. And our protein is only going to be 3 amino acids long, which isn't uncommon. So our amino acid that's only isoleucine, proline, and methionine is our protein. So now we made our protein, a methionine, proline, isoleucine. Okay? And that's translation. Okay? Turning DNA, transcribing it through the process of transcription into mRNA using RNA polymerase. Then we're going to take that mRNA, the messenger RNA, send it out of the nucleus into the harsh environment of the cell, and we're going to send it to the ribosome. The ribosome is going to start translating it into protein language, which is making it out of amino acids. And remember, we always have to start with AUG, methionine. That's the start codon. That means start. Let's start making our protein. If you don't have a methionine, don't make any amino acid. Skip it. Keep going until you get to AUG. Once you get to AUG, you're good to go. Start making your protein. You're going to keep making your protein on and on and on until you get to that last and final, where was it? UGA. Okay. If you get to UGA, that is your stop. Okay. That's your stop. And I'll move this down. Or if you get UAG or UAA, that's also stop.
At this time, the buses are starting to line up in the front of the building. Please make your way out to the buses. They will load quickly today and they will leave promptly. So again, students, start making your way out to the buses. Thank you.
Yeah. Okay. So, you need your start codon, you need your stop codon. That'll tell you how long your protein's going to be, how many amino acids you're going to make it out of. Okay? And that's what this pretty much tells you here. We take DNA, we use helicase to break it open, to break open that helix, and we use RNA polymerase to make RNA, to make the messenger RNA. But, tricky, remember, our binding rules change when you're talking about RNA, okay? So instead of A binding to T, A binds with U. So we replace all T's with U. Super tricky, okay? Super tricky. But if you notice on your codon sheet, there's no T's on here. So if you have T's and you're trying to figure out what amino acid you're trying to make, well, that should be ring, ding, ding. Oh, shoot, I need to change all those T's to U's, okay? That's a little hint there. Then we take this mRNA, we send it out of the nucleus, send it to our ribosome, so we can start attaching amino acids together, okay? That these amino acids are essentially hitching a ride on these T structures called tRNAs. And when we bind all these amino acids together, we're going to make a protein. And these proteins can have many different jobs, like signaling proteins. Ribosomes are actually made of proteins, so the protein builders are proteins, okay? Ribosomes are proteins, and they make proteins. Crazy enough. Structural proteins, supporting the cell, supporting cell structure, or changing cell structure. Enzymes, breaking things down, building things up. But the last thing I want to point out is often enough, a lot of these proteins will develop some sort of trait, okay? When you develop an external trait, something that can be observed, that's called a phenotype, okay? So, for example, look at me. I'm kind of pale. Not the most pale fella out there, but I'm pretty pale. I don't tan very often. I don't have much of this protein called melanin, okay? Melanin actually changes your skin color. And the more melanin that you have, usually the more dark you are, or the darker your skin tone is, okay? And that's a protein, okay? Proteins change how you look, okay? And so we start from the DNA all the way over here, and your DNA can change how you look by taking DNA, making RNA, and then translating that RNA with amino acids into proteins, and those proteins can determine how you look. Oh my goodness gracious, guys, we're almost there. Hot dog. Last but not least is enzyme function, okay? Brought them up before, but enzymes are proteins. Not all proteins are enzymes, but all enzymes are proteins, okay? And enzymes are proteins that break things down or build them up, okay? For example, catalase breaks down hydrogen peroxide. Lactase breaks down lactose. RNA polymerase builds RNA. DNA polymerase builds DNA, okay? Building or breaking. All enzymes end in the last three letters of ACE. So, like I said, RNA polymerase, DNA polymerase, lactase, catalase, invertase, all these enzymes end in ACE. Okay, if it ends in ACE, it's an enzyme. They speed up reactions. So, for example, hydrogen peroxide naturally breaks down in the presence of light. So if you shine light down on hydrogen peroxide, you're going to make water and oxygen. But if you add catalase to that reaction, that hydrogen peroxide is going to break down a lot faster, a lot more arrows, okay, a lot more speed. And we're going to produce a lot more water and oxygen. So they speed up reactions. Last but not least, I brought up this example earlier, starch. Starch is a compound made of 6 glucoses, and it's bound together. Starchy things that you guys eat are potatoes, french fries, etc., etc. And we need to break down starch. We need to break it down so it's in the form of glucose, so we can do things like cellular respiration and break it down so we can exhale CO2, we could exhale water, and we can produce that ATP. So to break this down, we have amylase, again, ending in ACE. It's an enzyme. Amylase is going to take this starch and it's going to grab onto it and start breaking it apart. So if I take this amylase, go right here, it's going to break that bond right in the middle. So if I take this and snap, it breaks it. And a glucose will end up flying out. If I do this right, I'm not doing this right. This glucose will go over there, it's gone. This one over here is going to break out and it's going to go and it's gone. Broke the bonds. We freed those glucose. And then this amylase is going to go and it's going to bind down to some more glucose on this starch and it's going to break that down. It's going to keep on going. And the more, let's say we have a lot of starch. I'm a starchy guy. I eat a lot of potatoes. Okay? If I eat a lot of potatoes, I might have a lot of amylase. And so I might have multiple amylases in my body. And these amylases, we're just going to start breaking down starch nonstop. The more enzymes you have, the more you can break down. Okay? Enzymes, structure, how they break things down, their proteins, okay, and an ACE. Okay, well, I think that's a pretty fun overview. I am sorry that the video is so long, but the longer it is, usually the better it is. I'm sorry with some of my antics of not being able to move some of the stuff around, but I think I got the message across, okay? Remember, be careful. Do not mess up DNA with RNA. RNA has U's, not T's, like DNA does. RNA polymerase causes transcription within the nucleus, versus translation is when that mRNA is sent to a ribosome to make a new structure completely, not genetic material like DNA or RNA, protein material. Okay? Hooey! If you have any more questions, just let me know. I think this covers a majority of everything that's going to be on the test and the review. I wish you guys luck. I'm going to try to provide this video as early as possible so you guys can just like watch it as that test is rolling on up. But with that being said, thanks for asking to make this video. I made it for Unit 4, 5, and 6, and one day I'm probably going to have to go back and make it for 1, 2, and 3. But because you guys have been asking, I got these videos out, and I think it's going to help other people. So I hope this helps. Let me know if you have any more questions in my email or in class or et cetera, et cetera. Thanks. Awesome. Have a good one.